Okay, it's a pleasure now to introduce Dr. Wen-Hann, who probably doesn't need much of an introduction. He is a medical geneticist coming from Boston Children's Hospital, and he is known as the natural history guy, who is going to be speaking to us about Angelman syndrome natural history study and all of your data. Everyone in the room and everyone who's watching, over 17 years of data, which is probably one of the largest natural history studies in the world for neurodevelopmental disorder. Thank you, Wen-Hann and team. He doesn't work alone, but he certainly represents in a really great way. So very excited to hear, and the community deserves this, so this is awesome. Do you want a mic, or would you rather just have that? That's okay?
Yeah. Slides. All right, so thank you, first of all, to Alison and the FAST team for inviting us to speak to all of you, and thank you for coming to this conference. I also want to thank Alison for her very gracious introduction. So I stand here before you really representing an entire team of really hardworking investigators, parents, participants who have made this study possible. And what I'm going to present to you really represents work that has been done by this amazing team of people over many years. So I want to start by just acknowledging, as I said, the very large team from all over the country, including our friends in Canada, who have made this possible. I will also say at the outset that even though this is a study based primarily in North America, we have had participants from all over the world, including Germany, Israel, Brazil, Ireland as well. So for those of you from countries where you do not have your own natural history study and would like to participate, please come to us. We'll be more than happy to see you. So what is a natural history study? You have heard a lot about the importance of biomarkers, and you're going to hear more about outcome measures. I think the juxtaposition between Jennifer's talk, my talk, and you're going to hear from Dr. Satwani's talk really fits all very nicely into a single package, and it all builds on data that we have collected. So many of you are already participants in this, so you know about this. It's an observational study. It's longitudinal. And I would stress the longitudinal component. And you will see in a minute that we really need families, parents to come back to us every year so that we can collect longitudinal data, not just data at one point in time. And this is over many years, of course. And we collect a mixture of what we call prospective data, i.e. we'll ask you questions that are things that just happened, as well as historical information, and the idea is to try to reduce recall bias. And the other important aspect of a natural history study is that you see the same set of investigators every time, wherever you may be seen. And that reduces the variability. And we have a very hardworking team. They are all out there at the desk table today, and they work on making sure that all the data they have collected are clean, curated, and trustworthy. And I'll also put a plug in, so the Global Angelman Syndrome Registry is really important. It serves its own purpose, and we collaborate. They complement each other, right? So the Global Angelman Syndrome Registry can reach a much larger population, but they may not be able to perform the sort of in-person assessments that we can perform. So I think both the Global AAS Registry and our natural history study have an important role to play. So I think you know the answer to this. Why do we need a natural history study? So you have heard a lot from this morning from Alison, then from Jennifer, about the importance of having natural history studies, having validated outcome measures. At an FDA workshop several years ago, there were speakers, representatives, both from industry as well as parent support groups, who talked about the importance of natural history study. So if you're going to conduct a clinical trial, you need to know if you see an effect on the clinical trial. Is that because of Angelman syndrome? Is that a side effect of the drug? Or what is it? Right? So natural history study data help us understand what is natural progression and what is the true effect of the drug. It also helps endpoints. We have had a great talk from Jennifer on endpoints, and a lot of the endpoints that we are using today really stemmed out of data that we collected through the natural history study. And as Emil Kakis, whom many of you know, is CEO of Ultragenics, who's one of pharma companies in this space, have said that natural history data is really what you need in order to develop and design clinical trials. But there are other reasons to do natural history study. You want to build expertise in investigators in order to conduct a clinical trial properly, in order to score, for example, your CGI that Jennifer referred to earlier. You need to understand Angelman syndrome, right? So the physicians conducting these studies need to develop expertise. And there's no better way to learn about any syndrome by seeing tons and tons of patients with that syndrome. So by participating in the natural history study, you're really helping us as clinicians to really understand your children, to learn about what Angelman syndrome really is in real life rather than from textbooks. It's also important to build an infrastructure. It takes time to build a team. And as you'll see in a minute, when I sort of explain to you how long it took us initially to set up a natural history study and build infrastructure, you realize that it's really important that we keep and maintain an existing clinical trials infrastructure at various sites across the country, across the world, so that we can launch clinical trials efficiently. So, and as I said, I think one of the mantras in medical school is we all learn from our patients. Our patients are the best textbooks. Your children, the participants, these are the best teachers that tell us what Angelman syndrome really is. And we have learned things through the natural history study that we did not know about, and we are just beginning to appreciate. And I think the other importance of natural history study is that it gives us the opportunity to really study a very heterogeneous group of population, right? So in any clinical trial, by necessity, you have to have participants who are stable, who have not had seizures for a while, who are on stable medication. So it's a very homogeneous group, and that may be very different from what real life is. And so we need to understand that. And the more important thing as well is that the natural history study goes on for a long time. So we have had 17 years of data that we have now accumulated. There are very few clinical trials that can afford to go on for that duration. So again, what we do in the natural history study really complements what all the companies are doing in their clinical trials. So the story really started in 2003, so actually 20 years ago, when the NIH decided that it would fund a set of consortia to study various rare disorders. And we were really fortunate that Dr. Art Burdett at Baylor College of Medicine applied for a grant and managed to secure a grant that forms the Angelman Syndrome, Prader-Willi Syndrome, and Rett Syndrome Foundation. The Angelman Syndrome component of this was led by Dr. Lindbergh at UCSD in San Diego. And although the grant was funded in late 2003, we did not, at the time when this grant was funded, there was no real clinical research infrastructure in the country for Angelman syndrome. So we had to look for investigators, psychologists, study coordinators, and we had to build this. And we had to start designing forms, designing the study, right? We had nothing to start with. So it really took a good 2 years and a bit before we were able to launch the natural history study. So you can see that we actually enrolled our first patient really only in beginning of 2006, even though the grant was funded at the end of 2003. And this really speaks to the importance of not just building, but maintaining the natural history study infrastructure so that we can launch trials. Today, we can launch clinical trials within 6 months of anyone coming to us. We could not have done that without the infrastructure that we have. So at that time, it was led by Dr. Burdett and then by Dr. Percy when the grant was renewed. And those are the study sites in the original natural history study, led, as I said, by Dr. Bird in San Diego. So this study ran for 8 years, during which this was the first natural history study of Angelman syndrome, and was a mixture of prospective data collection, as well as parent reports, And we saw each of these participants once a year at one of the 6 study sites. And we enrolled 302 subjects across all study sites. And I think many of the investigators today are in the room. Dr. Bacino is here, and Dr. Carson is here. They are all sort of part of this consortium. And give a shout out to Terry Gilbishel, who really promoted the natural history study aggressively and sort of made us do a lot of these things that we're doing today. So you can see that one weakness of this study was that we had relatively few adults in the study. In our first 302 participants, there were only 4%. And we also had a problem with dropouts, right? So a lot of people were very enthusiastic. They came for the first visit, and then, oh my God, I got to come back for a second visit, and then a third visit. And so we had a problem, right? We could not collect enough longitudinal data. We had really good data for sort of first visit, and then we started losing participants, as you can see there. So when we started, so there was a hiatus. We lost funding from the NIH, but we were very fortunate that the FDA agreed to fund us in late 2016. So we restarted this study in 2017. And by the time we started this new study, a lot had changed in the Angelman world. When we started the study in 2006, there were no industry partners. There was no one interested in developing therapies for Angelman syndrome. But by 2015, 2016, companies were starting to take notice, and they realized that Angelman syndrome was an eminently treatable neurodevelopmental disorder, and they started investing in it. So we started working with various stakeholders at that time, including various companies that were developing drugs for Angelman, or thinking of developing drugs for Angelman syndrome at that time, and we asked them, what would you like to see in this new iteration of the natural history study? Because one of our many guiding philosophies is that we want this natural history study to work for the parents, for all of you, as well as for the industry, right? Tell us what you need, and we will incorporate that into our natural history study. So one of the things we tried to do was, well, we had a very high dropout rate. Could we improve that? Perhaps by reducing burden. In the original natural history study, each visit was pretty long. It was like 5, 6, maybe 7 hours. And then it was, oops, what happened? Okay, and so we sort of said, well, can we reduce the burden by shifting some of the questionnaires that we would normally do in person to an online portal? So we did that in the hope that parents can then complete some of these questionnaires in person. And the other big major change was that, and this was really precipitated by the pandemic, we went to a hybrid model. So again, in the past, it was a full day visit, 5, 6, 7 hours. We said, okay, some forms can be done online, but in addition, can we do some of the questionnaires that requires sort of one-on-one interaction with the investigator over Zoom? And we did. So we now have a virtual and in-person visit, and we'll bring participants in to individual sites only for those assessments that cannot possibly be done over Zoom. So the Bailey is an example, a physical and neuro exam, for example, measuring head circumference, etc. Those things we had to do in person, but we managed to reduce the visit to just 2 hours, which really, I hope, greatly reduced the burden, because it basically allowed for parents to come in and just take half a day off work rather than a full day. We were again very conscious of making this natural history study relevant to the clinical trials that are going on. So we used the latest version of the Bailey. And again, we updated our Wen-Hann. When we started the first natural history study, ORCA had not been developed, but ORCA had been developed was developed in 2020, so we incorporated that in the natural history study. And I think this also points to the dynamic nature of how we want the natural history study to be. It's a living study. As things change, as outcome measures change, we need to be able to incorporate these outcome measures into the natural history study so that we can collect data. The other major thing we did was we want to expand access. So we went from 6 study sites to 10 study sites, including 3 in Canada. So we were really glad our friends in Canada were willing to participate, and give a shout out to FAST Canada and ASF Canada for supporting the study financially, and that has been very helpful to us. So these are the study sites across the country. And these are the outcome measures that were used in the in what we call the FDA-funded natural history study that started to incorporate outcome measures that some of the regulatory agencies wanted to see. Another innovation we undertook in this new iteration of natural history study was we say, well, instead of coming to Boston, to San Diego, to Chicago, and so on, can we go to the participants, go to sites where we see patients on site? So, for example, for the last 2 or 3 years now, we have been seeing patients and participants in the natural history study after conferences. So this year, for example, we are going to be seeing 5 participants on Sunday and Monday. After the FAST conference, we did that. We saw a good number of participants after the ASF conference in July, and we're going to do the same thing next year. We have also gone out to sites. We went to Austin, Texas earlier this year, and we also went to Princeton, New Jersey. That allowed us to see families that live close to the area. So we want to go to you where possible. There are limitations, of course, because this takes time, effort to organize a visit. So our funding from FDA ran out in 2022, and we were really fortunate that ABOM decided and agreed to fund us. So we now have the NHS 3.0, which is basically a continuation of the study with some modifications to make it relevant and up to date. And we changed some of the questionnaires. We decided that some of the questionnaires we used perhaps were no longer as useful. Some new questionnaires needed to be tried out, including CGI, for example. We introduced some sleep questionnaires. So the whole idea is to model this after a typical clinical trial. And we're going to try out at-home EEG. Some of you may have participated. If not, we may ask you to do so. And this is working with a company that will send EEG technicians out to your home to perform EEG at home instead of EEG in a hospital, and we hope that will be less traumatic and will allow us to get better data. So I'm going to go through this really quickly in the interest of time, but essentially, as I say, we want to share data with everyone. We are very open to sharing data. We want to support industry partners in design and development, and if anyone out in industry has some outcome measures they want to test, we would like to invite them to use us as a platform and to help us, and we'll help them get their observational studies up and running. These are the new measures that we're using in this outcome, in this natural history study. And these are the sites. So, where are we now? Well, these data are actually out of date as it is already. We now have, as of today, probably closer to 550 participants, if not more, in the natural history study. These are just data that have been entered into the database, so there's sort of sometimes a lag time. But you can see we have increased our number of adults. We still need more. It's still a relatively small percentage, but we have made some progress. And that's a breakdown of the molecular subclass, which I'm not going to. But you can see we still have a problem with attrition. We still have a problem with having people come back. So for all of you out there who have participated in the natural history study, please come back to us. Those of you who have not participated and are interested, as I said, we have a desk out there, and we have Lotus Lishti, Basheva Friedman, and Monica Chaffee out at the desk, and they will be very happy to enroll you, and you can participate in any one of the 10 study sites. You don't absolutely have to come to Boston. In fact, we probably don't need you to or want you to come to Boston. If you live really far away, you should go to your nearest site. So you can see that in this new natural history study, we have looked at some of the new measures. We are starting to collect outcome measures that many of these outcome measures will be used in clinical trials. So you have heard a lot about EEGs from the speakers before me. And really, the idea of being able to use an EEG as a biomarker started way back, more than 10 years ago now, when we started analyzing some of the early data in the natural history study, and we realized that there were markers that could be used. A lot of work went into it over the many years through collaborations with other academic groups, industry partners, etc. These are just, I don't expect you to be able to read or see any of these, except to say that there have been a huge number of collaborations across multiple groups. And you saw this slide earlier from Jennifer's talk, and we can now show that there is correlation between EEG delta power and cognition, and this is now utilized as a biomarker in the ongoing clinical trials. So this is real life being put in practice. And again, we have also shown changes in development and how it correlates with different subgroups. So we now understand a lot better about the different molecular subgroups in Angelman syndrome and how that impacts development. We can also see changes. And this is really important because we want to know how do the scores change over time so that we can compare and drug developers can compare the data that they have with what we have in the natural history study and ask the question, does that drug really work? Is it really improving scores over and above what you would expect without any intervention? Same thing with that. And same thing with Wen-Hann scores, Bailey scores. These are all measures you have used. Another major initiative that we have undertaken, that some of you have participated in, is to help us determine what is the minimal clinically important threshold for change, because just because you see a change in a score, is that clinically important? And we are going to develop developmental charts. This is under review right now, and eventually we're going to have to be able to tell you what to expect in terms of development for the kids. We've talked about measures. And finally, we just want to say that we have work in progress. We're going to try to develop growth charts, but we need more participants. We need more data points. We are going to try to develop a new clinical severity score that will be really, hopefully, really helpful for clinical trials in Angelman syndrome. And we want to understand adults with Angelman syndrome a lot more than we currently do. Finally, thank you to everyone who has supported this study financially and through other ways, as well as all the investigators, and finally, FAST, ASF, ABOM, both in US and in Canada, a very, very big thank you to all of you.
We have time for a couple questions before the break. Be one or two, if there's any. One thing I just like to make a statement, not a question, and I just want to make it really clear to the families why this is so important, is that everyone that joins the natural history studies and gives some data, it's a burden. It's a lot of work. It's a long day. It's likely some travel. It's not inexpensive to get there and to be there and to spend the time and take off of work. And then from that experience, it's hard to go back and do it again because you think, well, they have my data. But what's missing is to understand when you have data, it's one moment in time. And your child could have just had a really good day or a really bad day. And so it's hard to interpret that data if you don't have a second set of data over time to show how they're progressing with time. Because in a clinical trial, you have to show where are they today, and where are they in 3 months, and where are they in 1 year. And if you don't have that in the natural history study, longitudinal data, then your one-time data has dramatically less value. And so this is why we really encourage you, if you've gone once, at least just go one more time, because having longitudinal data makes the data set so much more rich and valuable. So I know it's a lot to ask, but we beg you, if there's one thing you can leave here with, sign up for search and rescue, and go back to the natural history study and do it at least one more time. And if you haven't done it yet, do it once and then do it again.
Thank you, Alison. So I cannot emphasize enough the importance of coming back to us, and ideally once a year if you can do it. As I say, we try to make it as easy as possible because a lot of these components can be done over Zoom. So it's really a Zoom call with one of the investigators.
This is Terry Jo from Combined Brain. You know, I'm a big proponent of natural history, but I want to maybe talk about how important it is for clinical trials. So my own son has been doing natural history for maybe, 15
Many
years?
Decades.
Decades. And we've gotten natural history visits in probably 4 different sites around the country, and we have data from all those years. So in fact, if in the future he has a treatment, a novel, amazing treatment that works, we might really be able to see that in him because we have all those years of natural history showing what he could do and what he couldn't do and when he plateaued. And if some new treatment should work, that might just be the evidence that changes everything.
Thank you, Terry Jo. And I think that's well said, and I think this is really the critical time for us to collect natural history study data, because now that we have treatment, now that there's a real prospect that we can really effectively treat Angelman syndrome, we need to know what the natural progression of the condition is. Otherwise, you will never know how effective a treatment is, especially long-term.
And these exist around the world. So this isn't just in the US and Canada, this is just this natural history study, but there's one very similar that's nearly mimicking in the UK, in Belgium, in Spain, coming in Latin America, in Australia. So they're happening all over the world, and we should encourage more countries to do it as well, and for them all to mimic each other.